16. That's the number of photoreceptor types inside the eye of a mantis shrimp. Humans have 3, and yet the mantis shrimp doesn't see more colors than you. It sees reality in a completely different way. By the end of this video, you'll understand 3 things that will permanently change how you think about vision, force, and what biology is actually capable of. Chapter: The Eye That Rewrote Vision Science. Let's start with the architecture. The mantis shrimp eye has what scientists call a mid-band, 6 specialized rows of cells stacked between 2 hemispheres. 12 of those 16 photoreceptor types are packed into this mid-band alone, covering wavelengths from deep ultraviolet at 300 nanometers all the way through visible light and into the far red at 720 nanometers. For reference, human vision covers roughly 400 to 700 nanometers with just 3 channels. Mantis shrimp cover over 400 nanometers of spectrum with 12 dedicated color channels, plus 4 additional channels dedicated purely to detecting polarized light. That's a paper published in Nature in 1999 by Justin Marshall and Johannes Oberwinkler, a landmark study that forced biologists to rebuild their models of animal vision from scratch. But here's where it gets strange. Those 12 color channels aren't stacked in a single eye pointing forward. Each eye sits on a movable stalk and can rotate independently on 3 axes. Each eye sees the world in 3 separate visual fields simultaneously, a feature called trinocular vision within a single eye. The mantis shrimp can judge depth with 1 eye alone. Humans need 2 eyes for that. The neural processing required to reconcile those independent streams every millisecond continuously is something neuroscience has barely begun to model. Chapter: The Punch That Breaks Physics. Now let's talk about the other thing mantis shrimp are famous for: the strike. These animals belong to the order Stomatopoda, and the smashing species carry a specialized limb called a dactyl club. When a peacock mantis shrimp strikes, that club accelerates at over 10,000 times the force of gravity. The tip reaches a velocity of 23 meters per second. To put that in context, a .22 caliber bullet leaves the muzzle at roughly 320 meters per second. The mantis shrimp's strike isn't supersonic, but it's in the same mechanical category as a ballistic projectile, generated entirely by biological tissue. The peak force recorded in laboratory conditions by Sheila Patek and colleagues at UC Berkeley, published in Nature in 2004, reached 1,500 newtons. Applied to a surface area the size of a pencil eraser, that's a pressure that shatters clamshells, crab carapaces, and, famously, aquarium glass. But the strike itself is only half the story. When that club moves through water at 23 meters per second, it creates a pressure drop so extreme that the water briefly boils at room temperature, a phenomenon called hydrodynamic cavitation. The collapsing cavitation bubbles release a secondary shockwave of force almost equal to the physical impact itself. The mantis shrimp hits its target twice, once with the club and once with collapsing vacuum. Chapter: The Paradox. 16 channels, less discrimination. Here's the part that surprised every vision scientist who heard it. Despite having 16 photoreceptor types and 12 dedicated color channels, the mantis shrimp is actually worse at fine color discrimination than you are. In a landmark study published in Science in 2014, Hannah Thoen and colleagues at the University of Queensland tested peacock mantis shrimp on a wavelength discrimination task. Humans can distinguish wavelengths as close as 1 nanometer apart using our 3-channel ratio comparison system. The mantis shrimp, with 12 color channels, could only reliably distinguish wavelengths about 25 nanometers apart. Why? Because they don't use their channels the same way we do. Human color vision is comparative. We perceive color based on the ratio of signals across 3 overlapping receptors. It's analog. The mantis shrimp, by contrast, appear to use each channel as a binary threshold detector. A specific wavelength either triggers a receptor or it doesn't. 12 channels, 12 yes or no answers. It's not a spectrum of hue. It's a color barcode lookup system. The brain doesn't blend signals. It matches patterns. This lets a mantis shrimp identify a color instantly and categorically, with no processing delay from comparison. It sacrifices discrimination for identification speed. Chapter: Engineering the Impossible Club. Now look at the structure that delivers 1,500 newtons without shattering itself. The dactyl club of a mantis shrimp has been studied extensively by materials scientists, and it has become a blueprint for impact-resistant composites. A paper by David Weaver and colleagues at UC Riverside, published in Science in 2012, found that the club is made of three structurally distinct regions. The impact surface is made of hydroxyapatite, the same mineral as bone, in a crystalline configuration that redirects crack propagation. Beneath that, a herringbone pattern of mineral fibers absorbs shockwaves. And surrounding everything, a helicoidal fiber structure, individual fibers wound in rotating layers like a spiral staircase, spreads tensile stress over a wide area before any single point can fail. Engineers have replicated this helicoidal structure in carbon fiber composites for military helmets and aerospace panels. A design that took 300 million years of evolutionary pressure to optimize is now being manufactured in factories. Chapter: What this means for your world. So what's the actual takeaway? The mantis shrimp isn't just a biological curiosity. It's a demonstration that there are multiple valid solutions to the problems of vision, force, and structural integrity. And that evolution has explored solutions our engineering hasn't matched yet. The real question isn't how many colors the mantis shrimp can see. It's this: how many problems in materials science, optical computing, and robotics are we still solving with three-channel human intuition? When the answer sitting in a reef tank is already orders of magnitude more sophisticated? What discovery about an animal has changed how you think about human biology? Leave it in the comments.